those who weren't here last week, we are trying to highlight the research that members of our department have been doing to show that bioengineering is an important and exciting discipline. I'm, as always, particularly thrilled to welcome current students and offer holders. And just a very quick bit of housekeeping. Please keep your microphones muted while Tom is speaking, and then we're going to have plenty of time for Q&A after the talk. I just want to briefly acknowledge Kemi for organizing all this. Thanks very much, Kemi. This is great. And then I don't want to keep you all much longer from why you're here. And it's my pleasure to briefly introduce Professor Tom Ellis, who is talking about baking our way out of the pandemic, which I'm sure is largely bait as a title, but maybe he will surprise me and actually talk a little bit about baking. Tom, the stage is all yours.
Okay, thanks a lot, David. Great. So it's lovely to be able to talk to you. There's probably a lot of people here who've never met me before or seen me, particularly offer holders. I'll be talking today about some of the work my research group and other research groups at Imperial have been doing to help out in the pandemic. We research a topic called synthetic biology, and that has become involved in some of the vaccine development work going on at Imperial College. And so this talk is going to give me a chance to introduce what synthetic biology is to some of you who might not know about it, and talk a bit about Imperial's work on vaccines and how some synthetic biology research in my group and some of the others has been helping out with the vaccine development work during the pandemic. So you see behind and on the slide the Bessemer building, and during the lockdown months of March, April and May, there are a few researchers beavering away in those labs. And you would have, if you've been seeing this series of talks, you would have seen that there are bioengineers, experts in working with materials and electronics and chemicals and nanofabrication to do things like make low-cost ventilators, sensitive diagnostics, and miniaturizing PCR testing to be able to test people very quickly for whether they have the coronavirus DNA. Now, this is what a typical bioengineering department lab looks like, with people having expertise in electronics and building things that can interface with biology and help develop things for the clinic. But playing around with actually the molecules inside cells and inside biology is also a part of bioengineering. Bioengineering now also includes engineering the components of life, particularly DNA, RNA, proteins, and cells. And that's really the research area that my group and several others in bioengineering department work on, and it's an area called synthetic biology. Synthetic biology, fundamentally, is about engineering cells to become new technologies that we can use, cells that can sense things and do things for us. And really the easiest way to think about synthetic biology in terms of a discipline is that it's kind of like computer coding, except that in synthetic biology, we work in programming DNA code, not writing a computing language that then instructs computers to put binary code into action into transistors and microprocessors. Instead, in synthetic biology, we look at DNA code, which is a bit more complex than binary. It's A, T, C, and G. And we think about different kind of words and sentences we can compose with A, T, C, and G that give us these DNA programs that can then be put inside cells to make them do new tasks. This is about getting the DNA sequence that encodes things like genes, rearranging and redesigning that code so that we can get the genes to turn on when we want and do things. This is quite an exciting research area, and it's also leading to a lot of applications. It's useful to think of synthetic biology and writing these DNA code programs a bit like writing apps that you can then put into a phone. If you think about it, what an app is it's a reasonably short amount of program code that can be added into an existing working system, a smartphone, and then it gives it a new functionality that can be useful and you can turn it on and off. And a lot of the time in synthetic biology, that's what we're doing. We're writing short lines of DNA code, relatively short, introducing them into a cell, which has its own DNA and does its own functions. And these extra lines of code make the cells perform a useful new task for us. So to give you some examples of work we do in my lab, we've designed apps that get cells to sense chemicals they wouldn't normally sense, things like hormones like melatonin, serotonin, or molecules like cannabinoids. And it takes us about 10,000 base pairs of DNA code, A, T, C's, and G's, to write programs that we'll put into cells that can do this work. We can also write apps that get cells to make things. So we wrote an app a few years ago, 20,000 bases of DNA code, a bit bigger here, 5 genes, that got cells to make the antibiotic penicillin, which was famously kind of stumbled upon by Alexander Fleming in Imperial's laboratory. Now, these lines of code are relatively small. They're equivalent to apps. If you think about apps, they are much smaller in coding and size and complexity than an operating system, like the operating system of a smartphone, which takes a lot longer to download when you're downloading an. Someone muted me. Hello, I'm back. Okay, so you think about the operating system, that is a big challenge, and we're also working on that in synthetic biology. The operating system of your smartphone may be several gigabases in size compared to the megabase size of megabyte size of your apps, and it's a similar thing with cells. Making a genome is much bigger. It contains thousands of genes split into chromosomes. But that's still something we're tackling in synthetic biology. My group and others have been working on making entirely synthetic genomes for cells. That's because that's such a bigger thing. It involves a worldwide collaboration where people are making from synthetic DNA new DNA code programs that encode all the functions of chromosomes and then bringing them together to make a whole genome. So if you think about the biggest scale of synthetic biology going on, we do that at Imperial. My group has made a 670,000 base pair part of a genome of an organism over the last few years with funding from EPSRC. This organism is called S. cerevisiae, and it's also the organism that we used as for those other apps, the cells that sense and the cells that produce antibiotics. This is the organism we really like to work with most in my group in synthetic biology. It's our magic ingredient, and it's also known as yeast, right? And so that's the link to baking. In fact, this form of yeast is called baker's yeast, sometimes called brewer's yeast, but mostly called baker's yeast. And we think it's a magic ingredient because it's a great cell to work with and does lots of things that make it really useful for synthetic biology. But it's for many centuries been thought of as a magic ingredient. Before it was discovered, it was always considered this kind of magic thing that happens in brewing and bread production, which gives us alcohol and gives us rising bread. There's a long history, many millennia, if you think about it, of people working with S. cerevisiae yeast, baker's yeast, because it gives us these wonderful products and a lot more, including things like chocolate and champagne as well. It's also very well used in biology and biotechnology research. It's a small organism that's very easy to work with. You can look at it under a microscope. You can put it through a life cycle very quickly to test out its fitness, but it's reasonably shares a lot of genes and functions with human and other animal genomes as well. So it's good for testing. And you can also ferment it to make products as well. So it's very useful in biotechnology. So even before this pandemic came along, people were bioengineering yeast for useful reasons for global challenges. And yeast synthetic biology, so that's engineering DNA code and putting it into yeast cells, has been at the forefront of synthetic biology's trying to solve global challenges, such as getting yeast cells that can break down the waste from agriculture and instead produce biofuels to fly aeroplanes with, rather than using fossil fuels, and yeast cells that produce components that you would normally find in meat, so that these can then be added into plant-based meat, so you can get things like the Impossible Burger, which seems to feel and taste like a real burger and even has this kind of a blood bleeding side to it, which comes from proteins made from engineered yeast. And they've even produced yeast that make spider silk, so instead of coming from spiders, and this can be used for new materials. So bioengineered yeast can help us solve many global challenges. How can it help out in a pandemic? Well, we already know yeast is very important in the pandemic, just based on looking at the supermarket shelves in March and April, when everyone was going crazy for some reason, of buying yeast, because people were interested in being able to make bread at home themselves, or maybe beer, and not be dependent on having to go to the shops to do so. This even led to Wired writing an article about the world running out of yeast for literally no good reason. And we can see how popular yeast was from Google Trend searches And the searches worldwide for yeast so far this year and have particularly ballooned at exactly the sort of same profile of the coronavirus cases in many countries, particularly in Europe. So there's a correlation perhaps there, and particularly sourdough baking. People culturing their own yeast at home to make sourdough bread has been a real trend and a bit of a meme online. In fact, so many people during this period when coronavirus was the worst number of cases were also making sourdough and culturing it at home. And some people even humorously speculated that the coronavirus was simply just a symbiont, a collaborating organism with yeast, and it was involved in getting yeast to spread around the world through nefarious mechanisms. So we don't think yeast is a bad guy. It's not helping out the coronavirus. Instead, it's something that's going to help against the coronavirus. So the question for this talk is whether bioengineering yeast can be the pandemic superhero, S. cerevisiae yeast. I think it can be, maybe not this sole superhero, and maybe not necessarily just for this pandemic, but maybe for the future. But I just want to point out something before I get into the talk, that while yeast might be a bit of a superhero in the pandemic, I'm not. A collaborator I've worked with in the past, Hannah Samad, wrote this very important piece early on in the pandemic to say that people really are, particularly the press and journalists, have this sort of narrative that they always go for, that they want to isolate this kind of male professor and how his team have been able to be a hero against the pandemic. And I think it's worth pointing out, Hannah's very right to point out, that this really isn't fair and true. Many people have children to look after at home, particularly young children, and can't become heroes. And it's really a bit selfish of the press to sort of put someone up on a pedestal and say their team did it. In most of the time, it's not them. It's the people who work in the labs rather than the professor. So please don't. I'm the person giving the talk today. My postdoc didn't want to do it. He's too busy doing the experiments. But please don't call me a hero. What I actually did in the pandemic was this kind of stuff. My wife works full time, so I was the one left with a small child. So I spent most of my time looking after her, trying to help her through learning how to do baking and crafting, and setting up large Zoom calls for 3-year-olds so that they can all sing songs to one another. And occasionally I managed to do some research. But I never went into the lab, and the work I'm going to present is nothing I can claim. Instead, it's the work of these important, great people. So Will Shaw, postdoc in my group, who I'm going to focus some of the talk on, what he's achieved, and the collaboration he's done with the other people you see on the slide here. Particularly Dr. Rochelle Orr, a really fantastic postdoc in Karen Polizzi's group, who are now in the chemical engineering department of bioengineering, and Jia Guo from Zhaoning Zhu's group in medicine, who are part of the people who have been working on vaccines for many years at Imperial. So these are the real people to celebrate, not me. I'm just the guy who's been stuck here in a shed in South London for the last 4 months and occasionally getting pestered by my daughter. These are the people who've been working hard, particularly the top row here in the lab. So how did we get involved in vaccines if we're doing yeast synthetic biology? Well, Karen, Rochelle, Jia, and Zhaoning, but not me and Will, are part of the Engineering and Physical Sciences Research Council's Future Vaccines Manufacturing Research Hub, which is one of 2 major vaccine hubs, research hubs in the UK, and this one's based at Imperial. And you can see the people involved in this project, and really what they're looking at, and this has been a center that's been established in the last few years, is how can we much more quickly produce vaccines when they're needed very quickly for threats as they arrive. So the coronavirus pandemic is something that this team of people have really risen to the occasion for, and it's very timely that Imperial had a research hub like this. But before I talk about some of the vaccine work that they're doing and how we've got involved in that, I just want to briefly talk a bit about biology and how vaccines work. When a virus or a bacteria infects you in your blood system or elsewhere in your body, some parts of the virus get recognized by immune cells. Particularly what happens is the outer surface of a virus, typically protein components, get kind of chewed up and get stuck inside cells such as antigen presenting cells, which you'll see here on the left here. And they then present bits of those proteins out on the outer surface for immune cells to come and inspect And if they look foreign, those immune cells will start to say, okay, this looks like the signature of something we need to attack. So they will trigger the production of immunoresponses, such as antibodies that are made specifically to target that little bit of protein that is being presented by the antigen presenting cells. That bit of protein is known as the antigen, and vaccines work by instead just providing antigen pieces to the immune system so that they can recognize what the threat looks like, but you actually don't provide the whole virus, because then that would lead to an infection. Now, this is an oversimplification. I saw this tweet the other day, and I completely agree with it, that the one thing that this pandemic has made clear is that no one understands immunology. It's incredibly complex, and Nick Lohman doesn't understand it, and I don't think I do either. So bear with me and don't ask me any difficult immunology questions, please, at the end. So, Imperial's vaccines. The most prominent one, which is really leading the way, is Robin Shattuck's self-amplifying RNA vaccine. And this is a very smart type of new vaccine that has never been seen before in human vaccine, but is really ready to go for this, because of our research hub, ready to go for this pandemic. It simply is a piece of DNA which encodes a piece of RNA that then goes into cells like the antigen presenting cells and then amplifies more copies of itself. And if you know about the central dogma of life, DNA makes RNA, and that makes protein, and antigens are the protein. So what we have is a little bit of DNA code that goes into the cells, amplifies to make many more copies of a piece of RNA, so that there's lots and lots of RNA, and they all then make lots and lots of copies of the antigen protein, and they can then be presented to the immune system, for the immune system to start to generate antibodies and T cells that recognize the antigen, which in this case will be the SARS-CoV-2 virus. Okay, this is a beautiful technology because it's so simple. It's just a short piece of DNA that encodes a piece of RNA that then just makes many more copies of itself. And this has been made using synthetic biology. The DNA can be assembled and harvested at large scale through E. coli synthetic biology. And the initial construction of candidate coronavirus vaccines using this method was just done by looking at the coronavirus DNA sequence online and then getting DNA synthesized by companies and sent to Robin's group to then build up to make these candidate vaccines. And this has gone very quickly. Despite this being a vaccine that's never been used before in human subjects, in 6 months, less than 6 months, from the DNA sequence of the coronavirus being released in China, Robin's team have now got to the point where this is in clinical trials and being injected into people. In fact, they estimate that the entire manufacturing of the first versions of these viruses in their labs only took a week or two. So a lot of the time instead has been spent on safety trials and animal trials before leading to the first people getting this vaccine in trials here in London. And this is an amazing success. And so Imperial have one of the leading vaccines in the world. We're in this group of 17 here that are in phase 1, lagging a little bit behind some of the others that are at the higher phases. But importantly, this vaccine is probably going to be one of the cheapest and easiest to distribute to the entire world. And that's why some impressive research grants have been given to Robin's group to consider how it could be, if it's successful, it could be rolled out all around the world. And part of that is the synthetic biology. E. coli can be grown up very quickly and easily to make millions and millions of copies of DNA. And only that DNA can then be very quickly distributed to the world. So that's Imperial's leading vaccine, which has some synthetic biology involvement, but not any involvement from my group. But Imperial also have another vaccine candidate down here that's been brewing in the background, kind of as a backup. And that's the project that we got involved in. This other vaccine project is something a bit more established as a vaccine. It's a type of vaccine called a virus-like particle. And this uses the fact that many viruses are assembled with a kind of protein cage around them so that they form a ball. You can kind of see that here in the slide on the left. The human papillomavirus, which causes cervical cancer, is a great case of this. It's this single protein, which when it gets expressed in the human cell, makes many, many copies, and they naturally self-assemble to make a big structure like a ball, And they will normally contain the viral genome inside, and this can then penetrate into the cells and infect the cells to make more copies. I've got a prop here, thanks to having a 3-year-old daughter. I have this, this funny thing here, if you see the image of me. And basically what you have is, you know, a structure where a single protein is repeated many, many times, and it makes a big cage. You can put the virus then puts its genome inside it, closes down, and it makes a virus particle. So a vaccine version of this simply is exactly the same, this protein cage, but without the genome in the middle, so no information to begin anything else. But the protein is there, and that will act as the antigen and trigger the immune system. And the most successful virus-like particle vaccine is called Gardasil, and it's literally the cage, this protein cage of the human papillomavirus, and it's given out to give a vaccine against cervical cancer with very, very high success rates around the world. So this technology of just this outer shell of a virus is a very useful potential vaccine. And how it can be made is quite simple. You simply just have to find the DNA that encodes this kind of self-assembling scaffold protein and express that in cells. So put that code into a cell and get it to make lots of copies, and it makes the scaffold. And if you want to change this VLP, this virus-like particle, so that it acts as a vaccine for different things, then you just have to attach onto this cage some other protein, which is the antigen. So a protein, for example, from coronavirus. And if you can literally fuse the two pieces of DNA code together and get a cell to express them, and if you design it right, you'll make a virus-like particle where this cage in the middle of the virus protein then presents on the outside the antigen that the immune system will recognize. So for SARS-CoV-2, the coronavirus, what antigen do we need to present if we're going to do a VLP? Well, that's straightforward, because there is one obvious thing to present, which is a protein called the spike protein. You can see it here in these beautiful images that people have produced over the last half year. It is this protein which sticks out of the virus, the SARS-CoV-2 virus, projects out and acts as the mechanism how this latches onto human cells and enters them. This huge protein, the spike protein, looks like this as it's binding down to something called the ACE2 receptor, which is present inside our bodies, in our lungs, in our nose, and in our bloodstream. And a little part of this spike protein, a little bit, juts out at the very bottom, called the receptor binding domain, which binds very tightly to the ACE2 receptor, and it aids the virus in then getting into the cells. This is really the lock and key mechanism by which this coronavirus attacks our bodies. It's very important, right? So this is the antigen we would want to present on a VLP. Either the whole thing or even just this thing would be ideal, because then if our immune systems can recognize this, they will produce antibodies which will grasp onto it, block it from being able to do its task. So this was recognized very early in the pandemic, in January and February, by Professor Zhaoning Zhu's group in medicine, who are part of the Future Vaccines Manufacturing Hub. And they quickly started developing a virus-like particle vaccine in human cell lines that used the spike protein as an antigen. So they take from the virus the spike protein, they take the region of this protein that is just the receptor binding domain, which we call the RBD. They then take the DNA that encodes that bit of protein and fuse that, genetically fuse that to a scaffold protein, which is what makes this virus-like particle shell. And then they can express that in a mammalian cell culture called HEC293 cells, and the virus particles will start to be made in those cells, and they can be harvested out of those cells and used in vaccine trials. And actually, their initial work suggests that this VLP vaccine works quite well in protecting mice from SARS, and that's a good model suggesting that this will work as a virus-like particle vaccine in humans. Now, the problem is mammalian cells are not a very scalable way to make virus-like particles. The Future Vaccine Manufacturing Hub produced this diagram you'll see here, which looks at the various different ways to make vaccines and how good they are at their speed, their cost, and the yield, how much vaccine you can make. The mammalian cell culture is the lowest speed way to make a vaccine. It's the highest cost way to make a vaccine, and it produces almost the worst amount of yields. RNA Robin Shattuck's virus has very high speed and very low cost, also produces quite a small yield. So what about some of these other options here? Well, a lot of them are microbes, and they offer good promise for making virus-like particles. So here are some microbes that you can make virus-like particles from. E. coli bacteria, which we use in synthetic biology, is the easiest for cloning DNA and very quick to work with. But unfortunately, E. coli naturally has toxins in it, so it means that it's very hard for you to take a virus-like particle made from E. coli and directly inject it into people. You would have to do lots of purification and checking first to make sure you're not going to make people sick from stuff left over from the E. coli. Two other yeasts are good candidates. Pichia yeast produces very high yields of products because it can be grown to a very thick density. It does unfortunately have some endotoxins you need to check for before you use something from Pichia and inject it into someone. Similar, another yeast called Hansenula is very good at folding proteins like these complex viruses up, but also has endotoxins. Baker's yeast, the one my lab works with in synthetic biology, is the cheapest to scale, and it's very safe. I mean, we drink it, we eat it, and it has no endotoxins. So theoretically, this would be the best thing to make a vaccine from very quickly, because it's very cheap and easy to grow this stuff, and it doesn't have to go through purification rounds and safety checks as much as the others would before you could deliver this into the bloodstream of people. And potentially in the future as well, if you really wanted to scale it up, don't forget that all around the world there are facilities and expertise for fermenting and making large amounts of product from yeast, and those are called breweries. So potentially one day in the world you could have a scenario where we could send bioengineered yeast to repurpose breweries for them to be able to quickly make vaccines on site and on demand. But probably we won't need to do that because there are existing proper clinical facilities for growing yeast to make virus-like particle vaccines, particularly in Vietnam and India. So I first talked to the Vaccine Manufacturing Hub last year, about November, introduced to Zhao by Karen Polizzi, and we discussed about ways we could optimize Baker's yeast, the organism we work with, using synthetic biology to be better at VLP production for vaccines that are needed in the future. We particularly talked about potential threats that may arise, the so-called disease X that people predicted might occur that would cause a pandemic. And who knows, we had no idea it was literally just around the corner, just a couple of months after we discussed this and thought about ways to begin a project on this. So we've been thinking about this a lot more, obviously, in my lab, and we have lined up a PhD student that will be co-sponsored between Zhao and myself, who will work on this in the future. But the pandemic hasn't waited, right? It's happened this year. So on the 17th of March, knowing that Zhao and his group and the Vaccine Hub's people were very, very busy, but knowing also that my lab was closing because bioengineering department was shutting down anything that wasn't essential, I quickly emailed Zhao and said, is there anything we can do to help? And he came back very quickly and said, yes, we have the VLP vaccine. We want to try and see if it can be manufactured in yeast. So this is where our involvement begins. My lab, Will Shaw in my lab, decided to take on the challenge of seeing whether the vaccine candidate that Zhao group was making in hex cells could instead be made more cheaply and quickly in Baker's yeast. Rochelle Orr from Karen Polizzi's lab would tackle it in Pichia yeast, which is better at scale up. And Rochelle actually came over to the bioengineering department labs and was based in the same lab as Will during the lockdown period. And Gia in medicine would look at Hansenula yeast. And actually, due to lots of issues with contaminations and people getting sick because they were based in a hospital, Zhao group, Gia really didn't make much progress on the Hansenula yeast. So I'll mostly be focusing on the progress we had in Baker's yeast and Pichia yeast using synthetic biology. The aim here really is that if the vaccine was going to go forward as one of the top candidate vaccines, this vaccine currently made in HEC293 mammalian cells, Imperial would like to have the ability to have the manufacturing transferred to instead be made much cheaper, much quicker in high yields from yeast cells. So that's what we're trying to achieve. We're the backup plan of the backup vaccine for Imperial. So don't worry if we fail. There's lots of other things Imperial are doing that can help. So here's the science. The spike protein is going to be the antigen for our virus-like particle. And it's either going to be the full spike protein, which is this very big protein, about 4,000 bases of DNA encode this. So it's about 1,254 amino acids, or just the very important receptor binding domain found at the end of this spike protein, which is much smaller, 234 amino acids, so about 700 bases of DNA is needed to encode that. So we want to put those on the outer surface of a virus-like particle, this kind of thing here, right? And Will decided to go for a very synthetic biology and very modular approach in our group. He decided to use design the system so that we produce the antigen so it can work with a modular technology developed by Mark Howarth, a previous collaborator of mine at Oxford University, who has a startup company called Spy Biotech. And what they do is they make at large scale, they manufacture ahead of time virus-like particles, making them from E. coli, like this kind of thing. But instead of having the antigen stuck on the outside surface, they have a piece of protein called the spy catcher domain, which is shown in blue here in this beautiful diagram. And I can pretend to put a spy catcher domain in blue on this virus-like particle here, because I'm in a shed, so I have some pegs, right? And so now they produce at scale a virus-like particle which has something called spy catcher on it. And if you literally you mix onto this particle a bit of protein that has another domain called spy tag. In the image here, it's this little orange piece of protein sticking out of the antigen in pink. Then the spy tag and the spy catcher covalently bind to each other, very high affinity, and now you stick antigen on the outer surface of your cell. So here's my purple peg, is my antigen. And if you see here now, I have a vaccine ready to go, a virus-like particle which is presenting an antigen, in our case, the spike protein receptor binding domain. And so it's a lovely modular system, and it means all we need to do if we're going to use Spy Biotech's VLP scaffold is make a large amount of this antigen protein with the spy tag attached to it. And so you can see Spy Biotech have a beautiful website that shows you how this works. You can see the virus-like particle here with now the antigen all around it, linked with these orange spy tags binding to the blue spy catcher domains. So simple. We only need to generate a very small DNA program in synthetic biology. It's shown here at the bottom. We need a piece of DNA which encodes a promoter to tell that DNA to be turned into protein. The first bit of the protein needs to be a little bit of amino acid sequence that tells the yeast cell to secrete this protein, so make it, but then put it out into the liquid for us to capture. Then the receptor binding domain, and then a tag on the end of the protein called the spy tag, so that it can then link up to the VLP scaffold. We take this DNA program, we transform it, so we stick it into the yeast cell, it goes into the nucleus, and DNA makes RNA and then makes protein, and that protein is made by the ribosome at the endoplasmic reticulum. It gets stuck into the endoplasmic reticulum, and the protein then gets taken through the Golgi body, through this secretion pathway, and then gets secreted out of the cells, normally with the secretion tag part chopped off, so that you just end up with the protein and its tag at the end. Great, so this is all we need to do. And Will Shaw in my group is an expert at modular DNA cloning. So this should be bread and butter for him to put these modules together to make this DNA program, choosing and designing the sequence online, picking and choosing appropriate modules. So once Will got started at this at the beginning of April, he made a whole variety of DNA constructs in the lab to then be put and tested into yeast. So we put promoters of different strengths, including inducible promoters. So these will respond to an input signal like a change in sugar and then make the protein, as opposed to just making it all the time. Different secretion tags and different tags at the end. So not just the spy tag that will help make the virus-like particle, but other options like his tag, which is one we can use to detect whether the protein is being made and secreted, or strep tag, which we can use to purify the protein if we need to do experiments from it. So Will spent a few weeks initially building up lots of different DNA constructs that will help us make this secreted antigen to go in the virus-like particle. And then we realized secretion was a lot harder than we'd hoped. So, for example, here's 4 different designs with different secretion tags that Will initially made, all under inducible promoters. And when he finally got to the point of trying to harvest protein out from outside the yeast cells, he saw nothing. So nothing was getting through the secretion pathways. We're not seeing a band in these positions here on this thing called a Western blot. You would normally hope to see a band like this one we see in the positive control. This tells us something is not working for us. Particularly, we simply think that this spike RBD protein we've designed is not being allowed to go through the secretion pathway of yeast. And actually, Will was telling us that the yeast seemed pretty sick as well, so there's something wrong in the design here. And so by early May, Will was looking a bit despondent on this project. And as the only person in our research group who was calling in back from the lab, which we'd all long forgotten whilst the rest of us were having our Zoom calls at home, Will seemed a bit like the guy left behind, like the guy from The Martian, struggling on in the lab on his own whilst the rest of us were all at home on our Zoom calls. So what was making Will have a tough time? Well, the answer is post-translational modification. And in the Future Vaccine Manufacturing Hub's schematic of what makes for a good production vehicle for a vaccine, we can see that there is a 4th thing involved at the bottom line here, which is the ability for different cells to be able to do the right kind of post-translational modification. And sure enough, the microbes, E. coli and yeast, are down at the poor end of the scale here compared to the other technologies. The post-translational modification that really matters, that causes trouble for us, is glycosylation. And so this is when, during the secretion pathway, proteins end up having sugars attached to them by enzymes that exist primarily in the Golgi body. And these sugars then kind of change the protein a little, and that can have big effects. So is the coronavirus spike protein glycosylated? The answer is hell yes, right? This is what the spike protein would look like if it was just protein, but it gets very glycosylated in the human cells, effectively cloaking it in a coat of sugar that makes it much harder for the immune system to deal with. Interestingly, though, the receptor binding domain at the top is not very glycosylated. So that means we do have a hope that receptor binding domains, this RBD region, still will make a good antigen for a vaccine. Recent research actually published this week in Cell has looked at, in Science magazine has looked at the different glycosylation profiles, the sugars that get put on the spike protein. And there's lots of different sugars. You can see in this fancy little diagram here, the different kind of sugars that polymerase and make these sugar chains we call glycans in the different positions around the spike protein. Now, these fancy little diagrams which show us the different kind of sugar glycans that build up at these positions are useful because they tell us the kind of sugars that we should expect to see if we're working in the human cells. But they also give us an idea of whether we're going to see differences in the sugar profiles in different organisms, because we know that in human cells, we see all of these different types of glycans made. You can see on this hand side of the diagram. But in other cells like insects, plants, and yeasts, we see different kinds of sugars appear at similar positions. And in particular, yeasts produce a very different kind of glycan. And our yeast, Baker's yeast, produces one called hypermannosylation, where it just goes nuts for this green type of sugar and makes lots of it in the positions. So our theory is that glycosylation of the receptor binding domain is causing the degradation of our protein as we're trying to secrete it in yeast. There are 2 sites in the RBD that are known to be glycosylated with these kind of profiles you see here that are a little bit similar to what you might see in yeast, but certainly not as complex as Baker's yeast's hypermannosylation. So we think these modifications with the hypermannosylation are going on inside yeast secretion pathway, and then yeast is seeing that and saying, this doesn't look right. It's chewing it all up and not wanting to secrete this. So over the last month or so, Will has gone back to our yeast cells and changed the genome, deleting certain things out from the yeast cell's own operating system to stop it from doing this hypermannosylation. So he's made a reduced glycosylation yeast strain with knocking out 3 genes. And now, sure enough, we are starting to see some production being some version, this design here, number 3, being secreted from our yeast cells. It's about the right size. Actually, it's slightly larger than just the protein size, which means it's probably still getting sugars attached to it, which might be a good thing, but might be a bad thing if they're the wrong kind of sugars. But at least it's not being degraded. And we can see in normal yeast, without this, we only see small, much smaller fragments of the protein being made, telling us that it is being degraded. Proteases, some kind of enzyme that exists in the Golgi body, are breaking things down. So Will, right now, this week, has added onto this a protease deficient strain, and he's been doing the cloning, and I think today he's doing this type of experiment, the Western blot, to see how well that works. And that's why when I told him, would you like to do the talk instead of me, he said, bugger off, I'm far too busy. I want to get this product up. I want to get this product made. So that's why I'm giving the talk on him. So that was Baker's yeast, and that's where we're at. Not fantastic. We're not at the point of having a vaccine made from Baker's yeast yet, but there's promise. Pichia yeast has been a bit more successful, largely thanks to its difference in how it glycosylates sugars. It's different to Cerevisiae yeast, Baker's yeast, because it doesn't do hyperglycosylation. It does high mannose, but it doesn't do lots of monosyllization. And the other reason this part of the project's been going very well is because Rochelle Orr is an awesome postdoc with amazing experience at Pichia pastoris. And so this is a brilliant project for her to be doing. And very early on in kind of April, May, she was already seeing that she could secrete the receptor binding domain, initially a truncated version, so we know it's getting chopped up a bit, but then starting to, through some changes in which Pichia strain she was using, she was able to start secreting out full-length versions of the RBD in detectable amounts. She did a lot of tests to see different ways she could make the antigen, both the RBD and the full-length spike protein, and had a very high throughput method for putting lots of different designs into different Pichia yeast strains, and then testing whether they're producing and secreting the antigen or not. And you can see some of her results here. She can test lots of different things out, and using this dot blot method, she can then find where she's getting large amounts of what looks like the right kind of antigen being made from different Pichia yeasts. So here we see trying to make the receptor binding domain from normal Pichia strain, trying to make the full-length spike protein. There's a few potential candidates here. And then more success with an bioengineered version of Pichia pastoris called Superman 5, which has been engineered to prevent hyperglycosylation, so having too many sugars attached to it. And so there's some good candidates here. And so Rochelle has even advanced to the point where the best full-length RBD candidates expressed and secreted from Superman 5 are then purified using strep tag and analyzed, and potentially then can be put into a system to make a VLP from Pichia yeast. So just to summarize this work, we're still ongoing on this. Both Rochelle and Will are still working on this. Pichia yeast can secrete the partially glycosylated RBD using Superman 5 strain, and that's been isolated and purified. Rochelle is trying to scale that up, purify it, to then start to take to an animal testing model. Back in Baker's yeast, we're not quite there. Will hopefully today will get good results that might show how we can get to the sort of same stage as Rochelle. But we think further strain engineering is definitely needed. Some yeast synthetic biology know-how is needed to get the yields up. Now, we know actually that yeast synthetic biology can help us get yields because some other researchers around the world have been using yeast synthetic biology in this pandemic. In this last 5-10 minutes, I'll just want to highlight some of the amazing stuff we've seen other people do with bioengineered yeast. One of them is a research group, the Bloom Lab in Seattle, which has used yeast surface display to put the receptor binding domain of the coronavirus on the outer surface of yeast. And actually, this uses the same secretion pathways we're trying to use, but when that protein, this antigen, gets secreted from the yeast, it immediately goes back and sticks on the outer surface of the yeast. So the fact that they've managed to get all versions of RBDs expressed well and secreted and put on the outer surface of the yeast shows that they're doing something right and we're doing something wrong. And we think that's that they're using a specific engineered yeast strain from past work that has been optimized for yeast surface display. So we're trying to get our hands on that right now for our project, because that might help. What they achieved in this research paper with yeast surface display is amazing. They were able to put the receptor binding domain, not just from this coronavirus, but from the previous SARS coronavirus and from other coronavirus, such as those found in bats and pangolins, express them all and put them on the outer surface of yeast and see how well they bind to the ACE2 protein to characterize how strong the interaction is in lots of different ways. And then they go even further than that, because yeast is so good at synthetic biology. They can use DNA synthesis and other methods to generate almost all of the possible 3,800 mutations that could occur in this RBD region of the spike protein of the coronavirus, and then see how different mutations, which may happen naturally as the pandemic's arising, can affect how this protein is expressed, how it binds to the ACE2 receptor, and how it will be bound by antibodies that might be used in vaccines, and whether some of these mutations might actually make it a worse virus and a virus that can evade the immune system. And so this is an amazing set of resources and information, all generated through some clever yeast synthetic biology methods by this team in the USA. Another amazing thing that yeast synthetic biology has been doing in this pandemic is actually making synthetic versions of the SARS-CoV-2 virus and other RNA viruses through a method known as reverse genetics. Baker's yeast, which we work with, is the best organism in the world for assembling synthetic bits of DNA together very quickly. And we've been using that in my lab for many years, and we've even taught how people can do that at a synthetic genome summer school in 2016. And one of the students on our summer school, Fabienne Labrousse, went away with that technology and now has come away with this research paper published earlier this year in Nature, showing that synthesized bits of DNA can be quickly assembled in yeast cells to make RNA viruses like the SARS-CoV-2 virus. They literally buy the DNA, they get it designed from DNA code, and a company makes these pieces of DNA, shoves them all together, puts them in yeast cells, and then they have a mechanism where this then assembles into the viral genome, which is only 30,000 bases of DNA, and then starts to produce virus particles when put into a mammalian cell. In fact, it only took this team 30 days when getting the coronavirus sequence first online in January to then be making the virus in their labs here. And the majority of that was actually just for the company to make the bits of DNA that were assembled. This is very impressive and shows the power of yeast. And I want to say it's also really scary that people can so quickly and frankly quite easily be able to get code from online and make a virus in a lab. And so we need to be wary of this. This is something we would call dual use technology, and it might be possible for having a good use. For example, we can synthesize versions of the virus with mutations in without having to wait for them to appear first and then test them, but also has bad uses that people might start making engineered viruses using this technology. So, baking and drinking our way out of a pandemic with yeasts. Well, lots of people have been baking during lockdown. Banana bread and sourdough are particularly popular, and a lot of people have increased their drinking. I know I have. It's kind of a bit like Christmas Day every day sometimes when you're at home. Can it help with the pandemic? Well, I think I've shown you various ways that yeast and bioengineered yeasts are helping out with the pandemic. Perhaps they're not going to be the ones to deliver the vaccines this time. Interestingly, probably the best candidate VLP-based vaccine, that's the kind of vaccine I've been talking about, will not be made from yeast, but actually is going to be made from a company, Medicago, who make it out of tobacco plants. Nicotina benthamia is a close relative to the tobacco people smoke, and actually is a very useful plant for making VLP-based vaccines, and that's one that's in quite advanced stages right now. Synthetic RNA made by E. coli and synthetic biology is part of two of the leading vaccines, Imperial and Moderna's one, and Imperial's one in particular is designed to be very low cost and quick to roll out. And probably the most advanced vaccine right now, produced by Oxford and AstraZeneca, is going to rely on those HEC293 mammalian cell cultures, which are really expensive and difficult to scale up, but thankfully a lot of people, a lot of governments and charities are putting money into that, so hopefully we can have this vaccine being rolled out later this year or early next year. I just want to say, though, that we're not going to stop this work. We think bioengineered yeasts need to be ready for the next virus, maybe something worse than coronavirus in the future, maybe something accidentally leaked from a lab, or even something designed by people for bioterrorism. We need to be prepared for all of these possibilities. And yeasts still provide the ideal platform for the lowest cost and highest yield production of VLP vaccines. We want to engineer the yeast cells to be good at that. And you can also see from the work of others that yeast synthetic biology allows for lots of rapid testing of mutations and for designing and building versions of the virus to see how they behave in very quick ways. So my time is up. I should end by thanking everyone who made this work possible. I didn't, like I said, I'm just been here stuck in my shed for months on end. Will Shaw has been the hero in my lab. Rochelle Orr has also been the hero in ChemEngine in my lab as well. And Jia and Zhao and Karen, part of the Future Vaccine Research Hub, have all been heroes as well, particularly Zhao and Jia with their group doing the work. And also thank EPSRC for sponsoring not only the research in my group, but the Future Vaccines Research Hub as well, and the Imperial College Bioengineering Department for allowing us to carry on working during lockdown in these labs and be able to get to where we are now. And thank you for listening as well.
Thanks very much, Tom, for this very interesting talk that I'm sure has raised many questions. So if participants would like to ask questions, you can either do so by raising your hand and I can call you to ask your question, or you can write your question in the chat and I will try and read it out to Tom, pronouncing all technical terms correctly. So please feel free to ask questions. Okay, there's a first question for you, Tom. Fantastic talk, Alex says, and he asks, how important do you think is it to have the correct glycosylation sequence on the RBD antigen?
It will help to have the correct, but I don't think it's essential, because as we see in some of the images of the glycosylation, the receptor binding domain, let me go rush all the way back here, the receptor binding domain region is probably the least glycosylated region of the spike protein. Oops. Oh, it's doing the thing. Sorry, zooming backwards and forward. And so compared to recognizing other parts of the virus, this is probably, it would be good to have the right glycosylation, but not necessarily essential. And we've seen groups, a group that I've collaborated with the past in Belgium and in the US, develop antibodies in vitro against the protein version of the RBD. And they have shown reasonably good enough strength to be neutralizing against the virus, even though they were evolved to recognize only the protein component and not the sugar component as well.
Thanks, Tom. There's another comment, not a question, but you're being told that you're a hero because you're at home doing the childcare. It's nice of you to acknowledge the lab. There's a question on Humanishi, and the question is, can we design cell organelles with the help of synthetic synbio?
Yes, and people have made kind of de novo organelles with synthetic biology using this kind of virus-like particle technology, where people are able to express proteins that naturally will self-assemble to make a kind of a cage, and then that provides an area that is separated from the rest of the cell. It provides a kind of like a miniature protein organelle where new reactions can occur inside that area that are separate from the rest and might have a different kind of chemistry as well. And you can the kind of proteins that are used in virus-like protein or particle production can be expressed in things like yeast cells to make new compartments.
So there's another question by Roshan, and the question is, you said that reverse genetics could make the virus, so do you think that this virus was possibly genetically engineered? That's the question.
I think no, not genetically engineered. I think we really don't know well enough, even the viral experts in synthetic biology, as to how you would design and modify existing viruses to make them better. Probably the virus has got very efficient at infecting humans by some circulation in a small population of humans that might have already had the antibody against SARS from previous exposures, and it is adaptive during time in those people. So, for example, a small community of people who might go into caves in Yunnan province in China to get product out, guano, and they're continually being exposed to the viruses that the bats have. They might have they might have generated antibodies over their lifetimes that mean that they have this virus, but it doesn't cause any problems for them, and then it adapts within that community. So you can imagine then if one of that community then goes outside, maybe goes to Wuhan for a holiday, it can then cause problems because it could end up in other people.
Thanks, Tom. The next question is by Maria, and the question is, what is it about yeast that makes it poor at post-translational modifications?
It just doesn't secrete too many things naturally, and that's similar both Pichia yeast and cerevisiae yeast. There's only a few proteins that it really wants to secrete. Therefore, it doesn't then need a whole plethora of machinery to work on those. So it's got a very narrow range of glycosylation. Whereas human cells, if you think about it, we're part of a multicellular system. We have to send stuff out to go talk to and bind and interact with other cells. So we secrete a lot of things out of our system. And so that means there's more machinery there and more evolution will occur to make different kinds of sugars go on it for different reasons.
Thanks. The next question, Thomas, have you made any studies or did you study at all the thermodynamic reactions in the synthesis of VLP?
Not us, no. I can't say about that, but there's people in synthetic biology and other areas who do a lot of work on protein design. And these proteins that are used to make VLPs, that are self-assembling proteins, are something people really have studied a lot, to the point where now experts in synthetic biology and protein synthesis can start to design de novo DNA sequences that encode amino acids that make peptides that are designed that they will naturally self-assemble. And all of that design process, a lot of it is based on thermodynamics. So there's definitely a thermodynamics component in that.
We've got a question by Ria. Ria, feel free to talk to Tom. I see that you raised your hand, so yeah.
Hi, thank you very much, Dr. Ellis. This was a lovely talk. So there has been quite a lot of hype about synthetic biology, especially during the recent years, I'd say. So what do you consider to be the main obstacles that we need to overcome to unlock the full potential of the field? And also, what regulations are there in place to ensure that these very powerful technologies do not fall in the wrong hands?
Okay. So I think the things we need to get a lot better at in synthetic biology are we need to understand biology better. And so we're very hopeful that lots of smart biologists and mathematicians can get together and start to do things like make on virtual simulations of the entire process of a cell. They've done that in a very small cell, and we think they're about to announce having done that for E. coli. And this now provides a sort of simulation environment that you can then test theoretical synthetic biology designs and changes in and see how they'll behave, which will hopefully make it less trial and error to do these kind of work we've described. Other things we would like to have a better ability to control mutation and evolution in cells. Evolution is basically the most powerful technology on Earth in terms of being able to self-optimize things, and it would be great for bioengineering to be able to harness that. And we'd just like to get better at building with DNA and writing with DNA at higher scale. So we want cheaper DNA from companies that make DNA for us. Now, what is there being done to prevent misuse, for example, of the technology to synthesize viruses? Well, there's a lot of community spirit in synthetic biology. We know each other quite well through education, particularly something called the iGEM community. People who've been undergraduates often enroll in a competition called iGEM where they will work together, and they really, it becomes kind of like there's a code of ethics if you've been involved with iGEM, and people kind of self-police in some way. And so that's really probably the most powerful thing you could do, is just have everyone interested in synthetic biology and using it on the same wavelength about what the risks are and what the benefits are and how to be responsible. The other thing is that the main DNA synthesis companies that you would order synthetic DNA from to make a virus have all signed up to analyze the sequences they get told to make and check if any of them look like virus sequences. And if they do, then they won't make it for you, and they will raise a flag and you will have to provide proper reasons for why you're doing this. And we hope that can partly limit, although it's not perfect, bad actors from trying to synthesize viruses.
Lovely, thanks a lot.
If you still have a couple of minutes, I'm going to ask you a few more questions because we have so many coming in. That's okay. Yeah. There was one question by Chris, and Chris wants to know basically how generalizable the process is. So once you have figured out to do it for one virus, what are the key things that will differ if you want to do it for the next virus, basically?
I don't think, I'm not a vaccine virus expert, but I don't think there will be that many things that are that different. I think what's so exciting about Imperial's future vaccine research manufacturing hub and the vaccines that we are following, VLP and self-amplifying RNA, and this current pandemic is we are basically doing all of this groundwork now that will theoretically allow much quicker kind of modules in the future. So as soon as we know there's an antigen that's going to be presented, we can encode part of that virus into a self-amplifying RNA. And if this works for coronavirus, hopefully it will work for other kinds of viruses and for bacteria as well. And then we can also do the same for VLPs, particularly with spy biotech's kind of base structure. We could just be putting different antigens. We could put multiple different antigens on. We could choose 2 and 3 and put them all on to make a bivalent or trivalent vaccine in one go. So really, we hope that this is going to revolutionize vaccines, both in the speed of their manufacture and then how quickly they can be rolled out as well.
I'm going to ask you 2 more questions. I'm just going to pick from the many questions that are being asked. Next question is whether it would be helpful to remove the glycosylation sites in the protein instead of lowering the glycosylation efficiency of the yeast.
Yes, that's right. And I think both Rochelle and Will, particularly Rochelle, have been looking at making mutations to this RBD and the full-length spike protein, changing some of those amino acids just slightly so that you still make the structure, but you no longer have the site on the protein that the sugar starts getting put on as it goes through the secretion apparatus.
Okay, then maybe as a last question is again related to glycosylation, and the question is whether glycosylated branches on RBD can cause extreme immune response or side effects that you're aware of.
Now you're asking me an immunology question, and like I said, no one understands immunology, and especially not me. So I'm not going to comment on that. But VLPs, not necessarily for this spike protein, are used. VLPs, even manufactured in yeast, are used in people. It's an established technology. So far, everything's looking good for Imperial self-amplifying RNA vaccine as well. So hopefully it's not an issue.
Great. Well then, thanks very much for this one. This was extremely interesting. So thanks for taking the time to give this talk. Thanks for all the people in the audience, in particular those who asked questions. And we have our next event on Tuesday, the 28th, when Professor Aldo Faisal is going to give a talk about what his group has been up to in the pandemic time. So thanks again, Tom, for this great talk. And I hope you all will have a good weekend.
Bye-bye, everyone. Have a safe weekend.